There's a sign-in sheet if you want. I'm not sure I can get better than that. Is this yours? Yep. Do you guys have any questions or comments or anything you'd like to ask Duran? If I'd looked, I would have known you.
Walker. You should be pretty good at it by now. All right.
I was trying to get it in one shot. I mean, you can get a nice picture in one shot, but you can't read it
it's too far
in one. And I'm trying to accommodate students and other people who aren't able to be here right now.
Yes.
You know what? What happened to my chat? Is it over there? No. Maybe I'll connect. If you have any questions and you want to say them, I will try to listen. If you want to chat them, I'll try to watch on my phone. I don't really need that. I don't actually know what I'm doing. I'm tilted a little bit. I'm not sure why. I don't think the cart is tilted, and I don't think the poster is tilted. I think it's just me and the camera tilted. I'm not sure where we're doing it.
I think it's this bit right there.
Yeah, this one. I don't think that it turns at this end, though I think it might turn here.
You get the gist of it. I can just run through it. It's not. So, I hope they can hear me.
Can you guys hear? Good.
For the two microbiological unknowns that we did in our microbiology class, we had to run a series of tests. We did our formation test, our metabolic tests, and basic gram stains. So we were presented with 2 tubes of bacteria, and we always start with a gram stain so we can learn if it's a gram positive or a gram negative, which, to just briefly describe that, is just the side of the peptidoglycan skin that's on it that goes around the cell. If gram negatives are very thick, so not a lot of gram stain can go into it, causes it a pink color, where gram negatives are thin, a lot of purple goes into it, you can see the purple coloring. So when we start with our gram stain for my unknown one, it was a gram negative. So I went straight into learning the morphology and the motility. My motility tests were inconclusive because our vials were very old. So I learned that it was a gram negative and it was rods, and I had no specific colony color. So we moved on. I did 3 plates, which the plates are just to be selective for specific bacteria, so it'll only grow certain bacteria. 2 of our plates are selected for gram negative only, so no gram positive bacteria will grow on them. So I used my 2 gram negative plates, which was an EMB and a McCogney. And those, by the color change, will tell me how much lactose it ferments and what is specifically on it. So for my unknown one, I had no lactose fermenting, so I went down to my metabolic. I had negative for urease, positive for citrate, negative for hydrogen sulfide, which is just the gases they produce when they go through respiratory cell breathing. And then for my fermentation, I had a weak lactose, I had sucrose and dextrose, which is just which specific sugars it creates, if it creates an acid or a gas. So with all those results, I have down here, I don't know if you can see it, a specific flow chart that you can follow where your results. For my unknown one, I had a specific bacteria called an enterobacteria Arogene? I don't know how to pronounce it. I do apologize. And then my tests for my second unknown were almost completely the same, except for I had a pink color, which everything else is exactly the same, which made it very difficult, and it is called serratia. And that is how you get 2 tests for 2 unknowns. You just, a bunch of fun.
I don't think I can
get any closer.
Yeah, well, I'm going to have to, like, stand on it to get
I don't want you to break your camera's neck. I don't want to take up all of your time.
No.
Fine. You got a lot of people to get through.
Thank you.
I ran through it. No, thank you.
It's nice to see you. I don't think I'm going to get a job as a movie cameraman anytime soon. Any questions or comments, folks? Okay. Thank you very much.
Thank you.
Yeah, wherever you like, whatever's good for you. I don't actually know what I'm doing, so mostly I gave this as an extra credit assignment to my students to attend, but a lot of them aren't able to physically be here, which is why we tried to set up a Zoom link thing. And really anyone who went to the school webpage could find the Zoom link. And there's another. So I'm gonna. Can you tell us a little bit about this?
Yeah, so my poster is about COVID's effects on our respiratory system. So as most of you know, what COVID-19 is, it's the virus that was discovered in 2019. And COVID-19 is going to be classified as an airborne virus. And if you look down here at the figure 1, you can see that what COVID essentially does is it affects the alveoli in our lung tissue. And what it does is it fills it with fluid. And this is the site of your gas exchange. So the more there's fluid in it, the less gas exchange you're getting, which is essentially less breathing. So, and then if you look over here at figure 2, you can see where it's black is where there is less oxygen rotating throughout your lungs. So over here is a healthy lung, and there's less black. And then over here is COVID scarred or COVID fibrosis. And all the alveoli in this person is going to be filled with fluid. Excuse me. And that is essentially the effects of ammonia as well. And ammonia is just essentially the filling of fluid in your lungs, causing inflammation. And then over here you have GGO, and essentially GGO is called ground glass opacity. And this is a CAT scan. And if you look over here where it's white, that is where the inflammation is in the lungs, and where it's just normal, there's no inflammation. And then if a patient with COVID goes to the hospital, they're essentially going to put them in the prone position, which is lying on your stomach. And what that does to the back of your diaphragm, your lung regions, it helps them expand open. And then for ventilation, the reason that some people on ventilation still end up passing away with COVID is because the lungs the ventilation is just filling the lungs with oxygen. It's not getting into the alveoli. The alveoli are still being filled with fluid. So overall, COVID-19, what it does is it causes inflammation in your lungs and fills it with fluid, which causes a decrease in the oxygen flow.
Thank you very much.
Yeah.
Does anyone have questions or comments? Thank you again. Hello. You can probably already tell that I don't actually know what I'm doing, so.
Oh, cool.
So there's the whole thing.
Did you taking pictures?
This is actually zooming the people on the northern website. The link includes a virtual link. So there are, I think, 4 people. And I'm trying to record this so I can put this on a YouTube link later.
Oh, okay.
Could you tell us a little bit about?
Well, I wanted to work with yeast and how it reacts with different kinds of sugars. This is actually an experiment by eScience that I kind of altered. During class, our results weren't really very accurate, so I wanted to return and try to see if I could get more, stable results. So what I did was I took different simple sugars, such as glucose and fructose, along with artificial sweeteners, such as stevia raw, Splenda, and sweetener. And then I mixed them with a yeast solution to see what the yeast would do. And then, in this case, the yeast would produce carbon dioxide. And with the different sugars, you can see that it produces different amount of carbon dioxide. For the simple sugars, down here, glucose and fructose, it's around here and here. For sucrose, which is actually a mixture of glucose and fructose, that is way higher. Something that I did find interesting is that the artificial sweeteners had much more of a difference gap between the results than the pure glucose and fructose.
Because it is able to mimic skin.
So that may mean there are some other things in the sweeteners that are reacting with the yeast that are not necessarily healthy. Now, that's not to say that sweet and low is not still a healthy substitute. It may react differently with yeast instead of with the human body.
What kind of yeast? I'm familiar with baker's yeast and brewer's yeast.
Oh, I just use normal baker's yeast.
I don't know the difference.
Me either, to be completely honest. I just thought there was one type of yeast until you mentioned that.
And they're also human yeast. People can get a yeast infection. And I believe those are all 3 different biological strains, but I am not a biologist. Questions, comments, anyone? People would be able to chat or talk, but I'm not sure I would be able to hear it or see it. Thank you very much, Cole.
Yes.
Is there someone on the back of that?
Yes.
Right, yeah, I've heard that. Thanks, Holly. How are you?
Yeah, I'm not going to be getting a job as a cinematographer anytime soon.
Having anything is great.
How are you, Holly?
All right, yourself?
These posters were actually presented at the West Virginia Academy of Science.
Cool.
These two are done by the anatomy and physiology students. And the last one is working with the technology. So Chris and Scott helped Tasha set up some technology to present the EKG data, which is what's being projected on the wall.
But I was just not expecting it. It was supposed to be, and I would assume that it would not live up to it.
Anything else? I don't want to leave you without
It's a relatively simple one compared to the bacteria. My friends different kinds of sugars, such as sweet and low and sweet row. I want to see how we would react or how we would talk. Specifically, I want to see the reaction between glucose and the state produce ethanol and carbon dioxide. And we can measure how much carbon dioxide is produced with this sort of mixture. What I've seen is that glucose and sucrose produce the least amount of carbon dioxide. It makes sense to me because glucose is very simple to produce. It's very light. And senior row is comprised of dextrose, which is just like glucose and so they perform relatively well. And this approach, which is the material side, perform the best, produce the most amount of carbon dioxide. Because it is a mixture of the simple sugar, glucose, and fructose. It's one thing that I've noticed is that the artificial like sugar packets show more deviation than your glucose. A lot of deviation. I'm assuming that's because there may be some other molecules in the packets that are not necessarily listed.
Okay. Yeah. And because that's not consistent packet to packet, you get the wider distribution.
What I'm not expecting at all is that sweetener was when people were talking about higher in sweetener in terms of that's the same as the sweetener is supposed to go lower calories, and I would assume that not form of level of sweeteners.
And I went back during the pandemic, harder to get. I went back to using sweeteners over regular sugar. There's a whole lot of difference.
I'm not necessarily sure if this means it is not healthy. It may just be the way it may just be the reaction is. And the healthy part is something I'm not looking at all.
Right.
There we go. It took me a while to realize why this was tipping. It's because this is bowing. This will be even better when we get David Attenborough to narrate.
The scariest thing I've ever heard about in school. Right. I'd be very fortunate. It's so fun to learn about this. You hear about them, you just don't Yeah, you don't make the connection. So do you want me to talk about it?
Sure.
Okay.
Love to.
I didn't know if that's what you were trying to get me to do or not.
Well,
Or do you want me to just like...
it's hard to see the whole thing and see the individual writing, especially on the screen. So I'm usually I'm usually giving them a broad view and then zooming in. Zooming in while we're zooming in.
Thank you. This is my favorite thing to do. It's kind of like a puzzle. I mean, you get two unknowns, and then you have to find what the bacteria is. So it can be difficult, it can be easy, but we run different tests so we know how to find it.
Focus better than that.
Yeah, he's doing
Go ahead.
Okay, we're good. Okay. So different things I did to start off, I did a gram stain. Gram stain is easy because you have to follow like these steps, which is like 60 seconds of crystal violet and 60 seconds of iodine. It's very, you have to like not mess up. You have a timer. But that way you can figure out if it's gram positive or gram negative. If it shows up purple, it's more positive. Both of mine were positive. And then if you zoom in on a microscope, it shows that they're little circles, so that means they're positive. That gave me a good indication of what it was, but I wasn't allowed to guess at that time. I had to keep running more tests, so I went ahead and grew it on an auger plate. The auger plates, right here, they are selective or differential. If they're selective, they'll grow on it. So these were selective to gram-positive because they grew gram-positive bacteria, and they're differential. So these two are the same place. They're called an MSA.
And this one was differential for the Staphylococcus aureus because it grew yellow. That meant it was fermenting lactose. Others don't. So this one was Staphylococcus epidermis. It didn't ferment lactose, so it shows up pink. So that gave you another clue. And then we also ran different metabolic tests. This shows acid production because it shows a yellow or an orange. They also have little rods in there. If they're up on the top, it shows gas production, but mine didn't have that. This is another one. It's called peptide iron auger. Pretty much what it is, it just shows you if it produces hydrogen. So my Staphylococcus epidermis did show it. And it also showed on this one as well. The only one that can indicate which one's different is the citric test. This just sees if it produces citrate, and if it shows blue, it does. So this one, Staphylococcus epidermis, did it. This is my unknown, and then I compared it with an actual one. And then this was your Staphylococcus aureus, and that's up there. So that indicated what that was. But it's definitely a big puzzle. You have a lot of things either you know and you had to redo, many tests ran, but I really enjoy doing this one more than the research because you're hands-on and it's more fun. So yeah, this one was fun. And it was kind of cool that I picked, because we had to pick which one we wanted, and I picked both Staphylococcuses, so it was kind of strange. But yeah, that's mine.
Great job.
Thanks.
Thank you.
You're welcome. Yeah, that one's awesome.
Right now, I'm this next week.
Push water into the
Great.
Because you're in the body naturally, so if there's too much cell, then so there's none of it. You know, because that gas and those increases, and we're trying to all I could have told you this one.
Yeah, sorry.
That's
Everybody's talking to this one. I know. Yeah, this is actually one I was talking about. I always would send it to you and I never did. Yeah, it's right here.
Hello.
Hello.
Yeah, I tried to tell him, I wanted to make sure that I got him. He texted her to know that it's mine, and
Well, Dr. Buchanan teaches math, right?
Yeah.
Yeah, I'm in your online algebra class.
Nice to meet you.
Good to meet you.
Just because of screen size, it's hard to see the whole thing and see individual words at the same time. Do you want to talk about?
All right.
If I'd looked at the name, I would have recognized it.
So, all things are made up of cells, obviously, and cells, they naturally have a certain amount of sugar and salt in them. So, what happens is if there's this process called osmosis, and what it does, it's when water will actually physically enter a cell to try to equalize the amount of sugar and salt and water that's on both sides of the membrane of the cell. So what I did was I took potatoes, because potatoes are plant cells, and I made different amounts of saltwater concentrations to see the size change over the different amount of salt. Like if you look here, I don't know if the camera's on.
Oh,
Right. But as you can see, here is 0% salt, which is just pure water. It actually increased the size of the potato because the water physically went into the potato to equalize the amount of salt on both sides. And once you get it goes shrink down to 20% on the next one, and it actually started to shrink a little bit, but not too much. And as you keep going, and the more salt you have in the water, the smaller the potato will shrink because the water will actually come out of the potato and into the solution to try to equalize the amount of salt and the salt and sugars on the other side.
I wonder if there would be much of a difference if you used whole potatoes. I wonder how much effect the skin has.
Yeah. I admitted that, but I'm sure it would affect it in some way. But it's like the same principle. If you eat a salty pretzel, you naturally will get thirsty because the salt enters your cells, and naturally your body wants to equalize the amount of water and salt on both sides.
So why do people brine turkeys? Doesn't that seem backwards?
Even people like Brian here just could like, at least.
I'm not drilling you. I'm just curious.
I mean, if I take like an educated guess, I would say the salt in the water would actually come out of the.
Yeah, but you don't want that to happen. I think that is what happens initially, and then if you leave it for a long time, it reaches an equilibrium.
I bet that has to do with temperature. Because mine's just all room temperature. There's no, because obviously for turkeys you're gonna cook them.
I wasn't trying to pick on you or anything. I have wondered that myself. We should get a culinary poster here next time.
Yeah.
Thank you very much.
You're welcome. Have a good day.
Oh, it changed. I thought it was all the same thing, and then it changed on me.
Obviously, it would be more of a 10%. But it wasn't a 10%. Because
That's the advantage when we plot this out. You don't have to plot it. It's the value of that line. Because it's evaporated. Now this is a little closer. So that's straighter. Wherever it crosses.
I ran all my time, so I didn't wish what it was. But I did.
Red line crosses and out. Yeah, well, I just that the rough and
Hello. How are you? Pretty good. Yeah, I changed.
help walk in the hours back then, now for breathing. Not in developed countries like the U.S. or the U.K., but undeveloped ones like
I don't actually know what I'm doing, so I'm faking it.
Is that audio on the screens?
There is. It's hard to hear from people with the background noise, and I haven't seen any chats. So it's intended to be interactive, but so far it's not terribly interactive. Do you want to talk about it, or?
If you have any questions for me, or if you just want to know about him, I can tell you about him.
I am not familiar with this person at all.
So he is a Jesuit poet. He was a priest, and he was born in 1844, and he died in 1889. One of his most famous works was God's Grandeur, which is a poem about how he believes God is in everything in the world that he's created, and his presence is in like nature and like beauty and everything natural. And over time, humans have come in and they've stomped on it, they've tore it apart and taken advantage of it. And in doing so, they have kind of forgot that God is there, and that's his presence, and that's how he sees the world. So a lot of this was influenced by what was going on in the 1800s. So a lot of inventions that were there were like the diesel engine, the automobiles, a roller coaster. So a lot of those things used oil and then the natural resources, of course, like wood for the roller coaster, and like metals and stuff that they had used for making of like the parts of the engines. So over that time, there was a lot of carbon emissions that were not filtered air. So just carbon emissions went into the air, and over the time, it has warmed up the atmosphere, causing a lot of global warming and caused a lot of insect, animal species, plant species to go extinct, and they're extinct. They try not to say extinct completely, because any efforts that would be to preserve that species would then be gone. Like there wouldn't be any funding to protect them. So he would just believe that, if we left everything alone, then it would thrive by itself, that we could live by itself. And he believes that nature, that even though there's all this destruction, that nature's going to turn around and come back and be fine. So, and I believe it. I think it's a good thing, because you see it in concrete, like when concrete breaks and you see weeds come through, it's nature coming back and saying, "I'm going to come get you."
Thank you very much.
Of course.
Forgive my awkwardness. I'm getting a little bit better, but I actually don't know what I'm doing, so.
Mine's a little bit longer than everybody else's, so I apologize on that end.
So
So I usually like to let them get an overview, and then I can sneak in a little bit. Do you want to talk about it, or?
Sure. This is about the polio virus and how it affects the central nervous system. The polio virus first starts out in the intestines. People can contract it through fecal contamination in water or food, and some of them it can get it through air droplets from a cough or sneeze. It's very highly contagious and incurable. If you happen to get it, you have to let it play its course. But there is some supportive treatment, like breathing ventilators. Like back in the 50s, it was the iron lung, and corrective braces to help with walking, things like that. There is a vaccine restricted strain that had a breakout in Congo. The only way that you can prevent that is through double vaccinations, because they say that even though you're vaccinated, you can still get that if you're in countries such as Pakistan and Afghanistan. There is 2 vaccines. There's a inactive one, which is the only one that the U.S. uses now since 2000, because the oral one has a active one that is weakened, and it has the potential of becoming back to being the natural state, which is causing polio, and those that are not vaccinated. Now, there is 3 types of polio virus. The only type that is still around is type 1. Type 2 was eradicated back in 2015 and 2016, and type 3 was eradicated back in 2019. Type 1 is still around. How it affects the nervous system is how, if the polio virus gets into the bloodstream, it enters the neurons, which causes the neuron to die, and it can't send signals to the skeletal muscles, which causes paralysis, and which could cause breathing difficulties and or death. Any questions?
You're slower than you, but you've talked about this before.
Mm-hmm.
I think I would be slower than you in any case.
Especially since I know the topic.
I haven't heard or seen any questions. Thank you very much, Joanna.
The next one's more for mine.
Oh, okay, cool. You know what? I'll
all of us look down together. Are you going to invest in it? Are you going to share your you know, whether it's a welding company or a roofing company, whatever it is, they got to have equipment. They got to check. They can see it and get started.
Okay, for this one, I had to identify 2 unknown microbes. My first unknown both of my unknowns were gram-negatives. I first started out with a gram stain to verify if it is gram-negative or gram-positive. Gram-negative is bacteria that show up pink when you stain it and under a microscope, and gram-positive is purple. Typically, majority of the time, the gram-negative is bacillus shape or rod shape, and a gram-positive is a coccus or circular shape. After I figured out that they're both gram-negative, I did all 3 plates, but I did a macaque plate that showed the different colors, and which also indicate if it ferments lactose or doesn't. My first unknown did ferment lactose, which had a pink coloration, which is over here. This indicates that it had a pink coloration. My second unknown did not ferment lactose, which it had a clear or colorless colonies. And then, my next one was to see if there was any fentanyl red test, which to see if it produced a gas or an acid. My first unknown had an acid and gas, and then my second unknown did not have, it just had an acid. So, and then I did a peptone iron test, which talked about to see if it produced hydrogen sulfide production. My first unknown did not, it was negative, and my second unknown did. So after looking at both of those, I had a hunch that the first one was E. coli and the second one was Salmonella. So I took the known E. coli and Salmonella and tested it side by side of my unknowns, which is this picture right here. And as you can see, it has the same coloration as the known, and which made it possible to figure it out that it was E. coli and Salmonella.
It's a little bit weird because sometimes people are coming in and going out. Some get disconnected and some have to work.
Yeah. And if I stutter, I apologize.
No, you were fine. Thank you very much.
Seems like they are looking at you as
Absolutely. You were in a meeting with Kelly, and they absolutely will come to the table and say, whatever we can do, just call us. Let us know what we do. It's an opportunity.
I read this. Montana and South Carolina. Now they're at a point where they discontinue the federal and swing because they're right. And I don't know if that's a good move or not.
That's a piece of the puzzle.
Yeah.
That's some of what we're experiencing here, but that's not all of it.
Oh yeah. But what you just said about there being a smaller labor pool, that's their way. They're not really thinking, they're thinking colors. They're not thinking about overall.
Yep. Business strategy. You've got to change. You've got to pivot. You don't have surplus labor. You've got to short it.
Thank you again, Joanna.
Hello.
Hello.
How are you?
Not a cinematographer.
That's okay.
Where's that?
They don't realize it yet, but they will soon.
There are
I know
things
the
that
blue probably doesn't count.
the blue isn't. There are things that sometimes tilt, and I don't know why they're tilting. See, part of the problem is this isn't entirely flat. It's slightly lumpy, and every time I move it, it gets on a different hump. So that looks okay. So we'll do that. So we've got an overview. And then zoom in so we can see a little better. I don't like to get too close because it ends up distorting. So I can.
So kind of keep a little bit of distance.
Well, I can electronically zoom rather than getting in close.
Mm-hmm.
Because if I get too close, then every time you're off to the side, it trapezoids it. Do you want to talk about this, or?
So what I chose to do is how COVID-19 affects your immune system. So with that being said, you have two different defense mechanisms. So you have your humoral, and then you have your cellulatory. And so what it is each one has their own different types of cells in them. And what it talks about is how when COVID is like ingested, and it goes like down into your immune system, how your cells fight off the virus.
First of all, hold on.
Thank you very much, Braylee.
Thank you.
Oh, yeah. Sure. This one?
Both of these are mine, so.
Okay. A better cinematographer would know what they're doing. I am not, and I do not. If you want to talk about it, you can.
So I did Staphylococcus aureus in the circulatory system of the heart. I chose the heart because a lot of complications come from this when it gets into your heart. Staphylococcus aureus causes endocarditis in the heart. And endocarditis is an inflammation of the inner lining of the heart's chambers and valves. There is acute and subacute endocarditis. Acute endocarditis develops suddenly and may become life-threatening within days. Subacute happens over a period of time, and it develops gradually, but it can also be very life-threatening. So, Staphylococcus aureus is a gram-positive, non-motile, and composed of a rounded-shaped populace clustered together, and that shows it in Figure 1. Staphylococcus aureus is considered a deep infection, which makes it very dangerous for your body and how it affects it. Staphylococcus first enters by a wound or a breach in your skin. It also can open through droplets. Once it gets into these openings, bacteria then move into your bloodstream and can go straight to your lungs, to your organs.